Hello and welcome to Nighttime Science. Tonight we are traveling to a realm that exists all around you, within you, beneath you, and above you, yet remains entirely invisible to your eyes. We are exploring the world of microbes, the smallest living creatures on Earth, organisms so tiny that millions could rest comfortably on the head of a pin, yet so numerous that they outnumber all other forms of life combined. Close your eyes for a moment and imagine the air around you. It seems empty, quiet, still. But within each breath you take, within each cubic centimeter of space, there float countless microscopic beings drifting like stars in an invisible cosmos. They are on your skin, in the soil beneath your feet, in the water you drink, in the food you eat. They have been here long before humans walked the earth, and they will remain long after we are gone. Microbes are not a single type of creature, but rather an entire universe of diversity compressed into the invisible spectrum of life. They include bacteria, which are single-celled organisms without a nucleus, their genetic material floating freely within their tiny bodies. They include archaea, ancient relatives of bacteria that thrive in some of the most extreme environments imaginable. They include microscopic fungi such as yeasts and protozoa, which are slightly larger and more complex but still invisible to the naked eye. Together, these organisms form the foundation of nearly every ecosystem on our planet, quietly performing tasks that make all other life possible. Most bacteria are so small that if you were to line up 1,000 of them side by side, they would barely span the width of the period at the end of this sentence. A typical bacterial cell measures between 0.5 micrometer and 5 micrometers in length, which means you would need a microscope magnifying hundreds of times just to see a single one clearly. Yet despite their infinitesimal size, bacteria are extraordinarily complex, each one a self-contained living system capable of sensing its environment, obtaining nutrients, reproducing, and even communicating with other bacteria around it. To put their size into perspective, consider this. If you could somehow enlarge a single bacterial cell to the size of a small car, a human being standing next to it would tower as high as Mount Everest. We exist on completely different scales, yet we share the same planet, often the same spaces, sometimes even the same bodies, living together in a relationship that has persisted for billions of years. Tonight, as you settle into comfort and let your mind wander through this gentle exploration, we will journey together through the microscopic realm. We will discover how these tiny organisms came to be, how they live, how they reproduce, where they thrive, and why they matter so profoundly to every breath you take and every moment you exist. We will explore bacteria that glow with their own light in the darkness of the ocean. We will meet microbes that can survive in boiling water and frozen ice. We will learn about the trillions of bacteria living peacefully within your own body, helping you digest food and protecting you from harm. This is not a journey of grand drama or sudden events. The world of microbes moves at its own pace, sometimes remarkably fast in microscopic terms, yet always invisible, always quiet, always unfolding in dimensions too small for our eyes to perceive directly. It is a world of patient processes, of slow accumulations, of partnerships formed over evolutionary time spans that dwarf human history. So let yourself relax, let the larger world fade away for a while, and let us descend together into the invisible, into the realm of the very small, where life exists in its most elemental and ancient forms, where the story of our planet truly begins. Let us begin our exploration by understanding the physical form of these invisible inhabitants. Bacteria, which make up the vast majority of microbes we will discuss tonight, are remarkably simple in their construction, yet this simplicity is precisely what has allowed them to thrive for billions of years. A bacterial cell is, at its most basic, a tiny sac of life. Unlike the cells that make up your body, which contain numerous specialized compartments and structures, a bacterial cell is essentially one open space enclosed by a protective membrane and a rigid cell wall. Inside this space floats the cell's genetic material, a single circular strand of DNA containing all the instructions needed for that bacterium to live, grow, and reproduce. There is no nucleus, no membrane-bound compartment to keep the DNA separate from the rest of the cell. Everything exists together in one continuous space, a design so efficient and so successful that it has remained largely unchanged for over 3 billion years. The sizes of bacteria vary, but most fall within a narrow range that defines the limits of what is physically possible for such simple organisms. The smallest bacteria measure about half a micrometer in length, hovering near the theoretical minimum size for life itself. At this scale, there is barely enough room inside the cell for the essential machinery of life. Ribosomes to build proteins, enzymes to facilitate chemical reactions, and that precious loop of DNA containing the genetic code. Any smaller, and there simply would not be enough space for a cell to function. On the other end of the spectrum, some bacteria can reach 5 micrometers or slightly larger, though these are the exceptions rather than the rule. Most bacteria you might encounter, the ones living in soil or water or on your skin, measure somewhere between 1 and 3 micrometers, small enough to slip through filters that would catch larger organisms, small enough to float indefinitely on air currents, small enough to inhabit spaces that larger creatures cannot even perceive. To help you visualize this scale, consider that a single human hair is roughly 70 to 100 micrometers wide. This means you could line up 20 to 100 bacteria across the width of a single strand of your hair, and they would all fit comfortably side by side in that narrow space. Or imagine this. If you were to collect all the bacteria living on the surface of your skin and compress them together into a visible mass, they would weigh only a few ounces despite numbering in the billions. Bacteria come in several basic shapes, each suited to different ways of life. The spherical bacteria, called cocci, are perhaps the simplest in form. Perfect little spheres that pack efficiently together and present the smallest possible surface area to their environment. Some cocci live alone, drifting as individual cells, while others cluster together in pairs, chains, or grape-like bunches, configurations that emerge naturally from the way they divide and whether they remain attached afterward. The rod-shaped bacteria, called bacilli, are elongated cylinders, sometimes short and stubby, sometimes long and slender. This shape gives them a larger surface area relative to their volume, which can be advantageous for absorbing nutrients from their surroundings. Many of the bacteria living in your intestines are bacilli, their elongated forms moving slowly through the landscape of your digestive tract. Then there are the spiral bacteria, twisted into gentle curves or tight corkscrews. These spirals, called spirilla if they form rigid curves or spirochetes if they are more flexible, can move through liquid environments by rotating their bodies like tiny drill bits, propelling themselves forward with elegant efficiency. The shape is not merely aesthetic, it is functional, allowing these bacteria to navigate through viscous fluids that would impede bacteria of other shapes. Each bacterial cell is surrounded by a cell wall, a rigid structure made primarily of a substance called peptidoglycan, which provides strength and protection. This wall is what gives bacteria their defined shapes and prevents them from bursting when they absorb water from their environment. Outside the cell wall, many bacteria have additional layers, capsules made of polysaccharides that help them adhere to surfaces or evade immune systems, or flagella, whip-like appendages that rotate like tiny motors to propel the cell through liquid. Inside the cell, despite the absence of compartments, there is remarkable organization. The DNA, though not enclosed in a nucleus, often occupies a specific region called the nucleoid. Ribosomes, the molecular machines that assemble proteins, are scattered throughout the cell's interior, ready to translate genetic instructions into the proteins the bacterium needs to survive. Some bacteria contain additional small loops of DNA called plasmids, which carry extra genes that can provide special abilities such as resistance to antibiotics or the capacity to break down unusual substances. This simple architecture, this elegant minimalism, is what makes bacteria so adaptable. Without the complexity of a eukaryotic cell, without the numerous specialized compartments that characterize the cells in your own body, bacteria can reproduce quickly, adapt rapidly, and survive in environments that would be lethal to more complex organisms. They are streamlined for survival, refined by billions of years of evolution into forms that waste nothing, carry no unnecessary baggage, and persist through circumstances that would extinguish almost any other form of life. As you rest quietly, imagine these tiny structures, millions upon millions of them, each one a complete living entity, each one carrying within its microscopic form all the information and machinery necessary to sustain itself, to respond to its environment, and to create perfect copies of itself. They are the most abundant life forms on Earth, and their simple beauty lies not in complexity but in efficiency, not in size but in persistence, not in visibility but in their quiet, pervasive presence everywhere that life can possibly exist. To understand microbes fully, we must travel backward in time, not merely decades or centuries, but billions of years, to an Earth that would be unrecognizable to you today. This was a planet without forests, without grass, without animals, without even oxygen in the atmosphere as we know it now. Yet this ancient Earth was not lifeless. In the shallow seas and along the edges of volcanic hot springs, the first microbes were already thriving, beginning the long process of transforming our planet into a world where complex life could eventually emerge. The oldest evidence of life on Earth comes in the form of structures called stromatolites, layered rocky formations created by ancient communities of cyanobacteria. These structures have been found in rocks dating back 3.5 billion years, placing the origin of microbial life in the earliest chapters of our planet's history, when Earth itself was less than 1 billion years old. The stromatolites formed when cyanobacteria grew in dense mats along the seafloor, trapping sediment and minerals between their layers, slowly building up mounds and columns of stone that recorded their presence like pages in an ancient diary. Even today, stromatolites still grow in a few rare locations around the world, particularly in Shark Bay, Australia, where the conditions mirror those of the ancient Earth. These living stromatolites are built by the same types of cyanobacteria that constructed them billions of years ago, organisms that have changed so little over geological time that they can be considered living fossils. To look at a stromatolite is to look at a structure older than almost anything else on Earth, a tangible connection to the dawn of life itself. Cyanobacteria were not merely the first microbes to leave clear fossil evidence. They were also the organisms that fundamentally changed our planet's atmosphere. Before cyanobacteria evolved the ability to perform photosynthesis, Earth's atmosphere contained very little free oxygen. Most organisms of that era lived in ways that did not require oxygen, extracting energy from chemical reactions that would seem alien to us today. But cyanobacteria developed a remarkable biochemical pathway. They learned to harvest energy from sunlight, using that energy to split water molecules and release oxygen as a waste product. This innovation, which emerged roughly 2.7 to 2.4 billion years ago, initiated what scientists call the Great Oxygenation Event. Slowly, over hundreds of millions of years, oxygen began to accumulate in the atmosphere. At first, this oxygen reacted with iron dissolved in the oceans, causing it to precipitate out and form the vast iron ore deposits we mine today. But eventually, the oceans became saturated, and oxygen began to build up in the air itself, gradually transforming Earth's atmosphere from one that would be toxic to us into one that could eventually support animals, plants, and all the oxygen-breathing organisms that followed. This was not a quick process. The transformation took place over timescales so vast that they challenge human comprehension. Imagine watching a rock erode, grain by grain, year after year, over your entire lifetime, and you would witness only the tiniest fraction of the time it took for cyanobacteria to oxygenate Earth's atmosphere. Yet these microscopic organisms, working without intention or awareness, rebuilt the chemistry of an entire planet simply by living, by reproducing, by carrying out the same photosynthetic process day after day, generation after generation, across hundreds of millions of years. The rise of oxygen was catastrophic for many organisms that had evolved in the oxygen-free environment of early Earth. Oxygen, which we think of as essential for life, was actually toxic to most of the microbes living at that time. The Great Oxygenation Event was, in effect, a mass extinction, eliminating countless species of anaerobic bacteria that could not adapt to the changing atmosphere. But it also opened up new possibilities, new ways of extracting energy from the environment, and eventually paved the way for the evolution of complex multicellular life, including eventually billions of years later, you. The cyanobacteria themselves survived, of course, and continue to thrive today. They still perform photosynthesis in oceans, lakes, and even in soil crusts in deserts around the world. Every breath of oxygen you take contains molecules that were produced by photosynthetic organisms, many of them cyanobacteria, continuing the work their ancestors began billions of years ago. In a very real sense, you owe your existence to these ancient microbes, to their accidental transformation of Earth's atmosphere into one that could support animals with lungs and blood and brains. Fossil evidence suggests that bacteria may have existed even earlier than 3.5 billion years ago, though the evidence becomes harder to interpret the further back we look. Some scientists believe that life may have emerged as early as 4 billion years ago, when Earth was still being bombarded by asteroids and comets, when the surface was hotter and more volatile than it is today. If this is true, then microbial life has persisted through nearly every major catastrophe in Earth's history. Asteroid impacts, volcanic eruptions on unimaginable scales, periods of global glaciation when the entire planet may have frozen over, and countless other disasters that wiped out most visible life forms. Through all of this, bacteria endured. They adapted, evolved, diversified, and filled every available niche, from the deepest ocean trenches to the highest mountain peaks, from scalding hot springs to permanently frozen Antarctic soils. Their ability to survive and thrive under such diverse conditions speaks to the remarkable flexibility of their simple design. to the effectiveness of their streamlined architecture, and to the power of evolutionary processes acting over immense stretches of time. As you lie quietly in the darkness, consider the depth of time represented by microbial life. Consider that bacteria were already ancient when the first animals appeared in the oceans 540 million years ago. They were ancient when the first plants colonized the land 450 million years ago. They were ancient when dinosaurs walked the earth, and they persisted unchanged when those dinosaurs disappeared. They are the elders of life on Earth, the organisms that have witnessed every chapter of our planet's biological history. And they remain with us still, invisible but omnipresent, carrying within their microscopic forms the legacy of billions of years of survival. One of the most remarkable characteristics of bacteria is the speed at which they can reproduce. Unlike animals and plants, which grow slowly, mature over months or years, and reproduce sexually through complex processes, bacteria simply divide themselves in half, creating two identical copies where there was once only one. This process, called binary fission, is elegance itself in its simplicity. When a bacterial cell is ready to reproduce, it first makes a complete copy of its circular DNA molecule. This copying process proceeds steadily around the loop of genetic material until there are two identical chromosomes within the cell. The cell then begins to elongate, growing longer as it prepares to split. A ring of protein forms at the cell's midpoint, gradually constricting like a drawstring being pulled tight. As this ring tightens, it pinches the cell membrane and cell wall inward, creating a deepening groove that eventually meets in the middle, separating the one cell into two. Each daughter cell receives one copy of the DNA and roughly half of the parent cell's cytoplasm and molecular machinery. Within moments of separation, each new cell is already a fully functional bacterium, capable of absorbing nutrients, growing, and preparing to divide again. There is no juvenile stage, no period of maturation. The moment of division is the moment of full functionality. The speed of this process depends on conditions, particularly the availability of nutrients and the temperature of the environment. Under ideal conditions, some bacteria can complete the entire cycle of growth and division in as little as 20 minutes. The bacterium Escherichia coli, commonly known as E. coli, which lives in your intestines and has been studied more thoroughly than perhaps any other organism on Earth, can divide every 20 to 30 minutes when given optimal nutrients and temperature. Let us pause for a moment to consider what this means. If a single E. coli bacterium were placed in an environment with unlimited nutrients and space, and if it divided every 20 minutes, then after 20 minutes there would be 2 cells. After 40 minutes there would be 4. After 1 hour there would be 8. After 2 hours, 16. The progression continues exponentially, doubling with each generation. After 10 hours, with 30 generations of division, that single bacterium would have produced over 1 billion descendants. After 24 hours, the number would exceed what we can easily comprehend in everyday terms. This is the power of exponential growth, a mathematical reality that plays out in the microscopic world with profound consequences. Of course, such unlimited growth never actually occurs in nature. Bacteria quickly exhaust their nutrients, produce waste products that become toxic, or encounter other limiting factors that slow their reproduction. But this potential for rapid multiplication explains why bacteria can colonize new environments so quickly, why bacterial infections can develop with surprising speed, and why bacteria can evolve and adapt to changing conditions much faster than larger organisms. The rapidity of bacterial reproduction also means that mutations, random changes in the DNA sequence, occur frequently in bacterial populations. Most mutations are neutral or harmful, but occasionally one arises that gives a bacterium some advantage in its environment. Because bacteria reproduce so quickly, a beneficial mutation can spread through a population in a matter of days or weeks, allowing bacteria to adapt to new antibiotics, new food sources, or new environmental conditions with remarkable speed. Not all bacteria reproduce at the same rate. Some, particularly those living in nutrient-poor environments or extreme conditions, may divide only once every few hours or even once per day. Slow-growing bacteria tend to be more efficient with their resources, more careful in their cellular processes, and often more resilient to stress. They represent a different survival strategy. Rather than overwhelming through numbers, they persist through patience and efficiency. The act of division itself is nearly instantaneous once the constriction begins, taking only a few minutes for the physical separation to complete. But the preparation, the growth phase where the cell accumulates enough mass and resources to split successfully, takes much longer. A bacterium must essentially double its entire contents before it can divide. It must copy its DNA, synthesize enough ribosomes for 2 cells, build enough cell membrane and wall material for 2 cells, and accumulate enough of every enzyme and protein to ensure both daughter cells will be viable. This process of preparation, this gathering of resources and molecular machinery, is where most of the generation time is spent. The actual moment of division is merely the culmination of a long period of quiet growth, invisible even under a microscope, as molecules are assembled one by one within the cell's interior. As you rest here in the darkness, bacteria throughout your body and in the environment around you are engaged in this ancient process. Some are growing, accumulating the resources they will need to divide. Some are in the act of division itself, their cell walls pinching inward to create 2 where there was 1. Some have just divided and are already beginning the cycle anew. It is a process that has continued uninterrupted for billions of years, a rhythm of life at the smallest scales, proceeding whether we notice it or not, whether we are awake or asleep, whether we exist or not. The bacteria were here before us and they will be here after, dividing and dividing and dividing, perpetuating themselves through the simplest and most elegant means evolution has devised. While most bacteria thrive in moderate conditions in temperatures and environments that we would consider comfortable, some microbes have evolved to survive and even flourish in places that seem utterly inhospitable to life. These organisms, collectively known as extremophiles, challenge our understanding of the limits of life itself. Consider the thermophiles, bacteria that live in water so hot it would scald your skin instantly. These remarkable organisms thrive in the hot springs of Yellowstone National Park, in deep-sea hydrothermal vents where superheated water erupts from cracks in the ocean floor, and in other volcanic environments around the world. Some thermophiles grow best at temperatures between 70 and 80 degrees Celsius, hot enough to brew tea. Others, called hyperthermophiles, prefer even more extreme heat, with some species thriving at temperatures exceeding 120 degrees Celsius, well above the boiling point of water at sea level. How do these bacteria survive such temperatures? The answer lies in specialized proteins and cellular structures that remain stable when the proteins in your own cells would denature and fall apart. Thermophilic proteins have subtle differences in their chemical structure that allow them to maintain their shape and function even as thermal energy causes their molecules to vibrate intensely. Their cell membranes contain special lipids that remain fluid and functional at high temperatures where normal membranes would simply melt. In the hot springs where thermophiles live, you can often see their presence in the vibrant colors that ring the pools. Different species of thermophilic bacteria produce different pigments, creating bands of orange, yellow, green, and red that shift with the temperature gradient as you move away from the hottest central waters. These colors are not merely decorative. The pigments protect the bacteria from intense sunlight and may play roles in their unusual metabolisms. At the opposite extreme, psychrophiles thrive in cold environments that would seem equally impossible for life. These cold-loving bacteria inhabit the permanently frozen soils of Antarctica, the depths of polar oceans, and ancient glacial ice. Some can reproduce, albeit slowly, at temperatures well below 0 degrees Celsius. They accomplish this through proteins and enzymes that remain flexible and functional in the cold, and through the production of antifreeze compounds that prevent ice crystals from forming inside their cells and tearing their delicate membranes. Scientists have discovered psychrophiles living in ice cores drilled from deep within glaciers, where they have been frozen for hundreds of thousands of years. When these bacteria are carefully thawed in laboratories, some resume their metabolic activities as if no time had passed at all, demonstrating an almost incomprehensible patience and resilience. Then there are the acidophiles, bacteria that thrive in environments so acidic they would dissolve metal. These organisms live in sulfur-rich hot springs where the pH can drop below 1, approaching the acidity of battery acid. They also inhabit acid mine drainage, toxic runoff from mining operations where the water is bright orange or red with dissolved metals. To survive in such corrosive environments, acidophiles maintain their internal pH at near neutral levels through specialized pumps and barriers that prevent the external acid from destroying their cellular machinery. Alkaliphiles represent the opposite adaptation, thriving in extremely basic environments with pH levels above 10. These bacteria live in soda lakes, bodies of water saturated with sodium carbonate, where the water feels slippery and soapy to the touch. Like acidophiles, alkaliphiles must carefully regulate their internal chemistry to prevent the harsh external environment from disrupting their cellular processes. Some bacteria have adapted to withstand enormous pressure, living at depths in the ocean where the weight of the water above would crush a human instantly. These barophiles or piezophiles inhabit the deepest ocean trenches, places where pressures exceed 1,000 times atmospheric pressure at sea level. Their cell membranes and proteins have evolved to function normally under this crushing weight, incorporating structural features that would make them unstable at the surface. Halophiles love salt, thriving in environments like the Dead Sea, or salt evaporation ponds where the salt concentration is so high that most organisms would quickly die of dehydration. These bacteria accumulate compatible solutes inside their cells, organic compounds that balance the osmotic pressure from the salt outside without interfering with their biochemical processes. Some halophiles produce pink or red pigments, giving salt ponds their distinctive colors visible even from space. Perhaps most impressive are the radiation-resistant bacteria like Deinococcus radiodurans, which can withstand doses of radiation thousands of times higher than would be lethal to humans. These bacteria possess extraordinary DNA repair mechanisms, able to reconstruct their shattered genomes even after intense radiation exposure breaks their DNA into hundreds of fragments. Scientists have found these organisms living in nuclear reactor cooling pools and other highly radioactive environments. What unites all extremophiles is their demonstration that life is far more flexible, far more adaptable than we once imagined. The conditions we consider normal, the moderate temperatures and neutral pH and comfortable pressures of our everyday environment, are merely one small portion of the spectrum where life can exist. Extremophiles expand that spectrum dramatically, showing us that life can find ways to persist in nearly any environment where liquid water exists, and sometimes even in places where water is frozen solid or exists only as vapor. As you breathe slowly and let your mind wander through these extreme environments, imagine these bacteria going about their lives in boiling water, in frozen ice, in acid, in salt, in darkness thousands of meters below the ocean surface. They are not suffering or struggling, they are thriving, perfectly adapted to conditions that would kill you in moments. They remind us that life is not fragile but remarkably tenacious, not rare but almost impossibly persistent. Finding ways to exist wherever chemistry allows, pushing against the boundaries of what we thought possible and quietly redefining those boundaries with their mere existence. We have explored bacteria in ancient rocks, in boiling water, and in frozen ice. But now let us turn our attention closer to home, to the bacteria that live within and upon your own body. You are not merely a single organism, but rather a walking ecosystem, a living habitat for trillions of microbes that call your body home. The human microbiome, as this collection of microorganisms is called, consists of bacteria, fungi, viruses, and other microscopic life forms that inhabit your skin, your mouth, your airways, and most abundantly, your digestive tract. Scientists estimate that a typical human body harbors approximately the same number of bacterial cells as human cells, roughly 30 to 40 trillion of each. Some earlier estimates suggested bacteria outnumbered human cells 10 to 1, but more recent research has refined this ratio to approximately 1 to 1. Still, because bacterial cells are so much smaller than human cells, all the microbes in your body together weigh only about 200 grams, less than half a pound. The vast majority of your resident bacteria live in your large intestine, the final section of your digestive tract where water is absorbed and waste is prepared for elimination. Here, in this warm, dark, oxygen-poor environment, bacteria exist in concentrations that stagger the imagination, reaching densities of 100 billion cells per gram of intestinal contents. If you could somehow collect all the bacteria from your colon and examine them, you would find representatives of thousands of different species, though most belong to just a few major groups. The dominant bacterial phyla in the human gut are the firmicutes and the bacteroidetes, which together typically account for more than 90% of the gut bacterial community. Firmicutes include many species that help break down complex carbohydrates and fibers that your own digestive enzymes cannot process. Bacteroidetes are similarly skilled at degrading plant polysaccharides, working to extract nutrients from the plant matter you consume. Together, these bacteria act like an external digestive organ, breaking down food components that would otherwise pass through your system unutilized. Other important groups include the actinobacteria, which include species that produce vitamins your body needs, and the proteobacteria, a more diverse group that can increase in abundance during digestive disturbances. Each person's microbiome is unique, shaped by genetics, diet, environment, medications, and life experiences. The bacterial community in your gut is as distinctive as a fingerprint, a signature of your individual biology and history. These intestinal bacteria provide services you might not realize you depend upon. They synthesize certain B vitamins and vitamin K that your body cannot make on its own. They train and regulate your immune system, helping it distinguish between harmless substances and genuine threats. They produce short-chain fatty acids as metabolic byproducts, which your intestinal cells use as an energy source and which help regulate inflammation throughout your body. They compete with potentially harmful bacteria for space and resources, preventing pathogenic species from establishing themselves in your gut. The relationship between you and your gut bacteria is one of mutual benefit, a symbiosis that has evolved over millions of years. You provide them with a stable environment and a steady supply of food. They, in turn, help you digest that food, produce essential nutrients, and protect you from disease. Neither party consciously chose this arrangement. It emerged through evolutionary processes as humans and bacteria coexisted, each adapting to the other's presence until the relationship became essential to both. Your skin hosts its own distinct bacterial communities, different species adapted to different regions of your body's surface. The bacteria living on your forearm differ from those on your face, which differ from those in your armpits or between your toes. Oily areas tend to host different species than dry areas. Moist, warm environments like armpits support different communities than exposed, cooler skin. These skin bacteria help protect you by occupying space that might otherwise be colonized by harmful species, and by producing compounds that inhibit the growth of pathogens. Even your mouth contains a complex bacterial ecosystem with different species colonizing your teeth, tongue, cheeks, and gums. Some of these oral bacteria form dental plaque, biofilms that can contribute to tooth decay and gum disease if allowed to accumulate. Others are harmless or even beneficial, producing substances that inhibit harmful bacteria or participating in the initial stages of food digestion. The composition of your microbiome changes throughout your life. Infants acquire their first bacteria during birth, receiving an initial inoculation of microbes from their mother. Breastfeeding further shapes the infant microbiome, as breast milk contains not only nutrients but also specific sugars that feed beneficial bacteria. As you grow, your diet, environment, and experiences continue to shape your bacterial communities. The microbiome of an elderly person typically differs significantly from that of a young adult, reflecting decades of accumulated changes. Disruptions to the microbiome, particularly from antibiotics, can have profound effects. Antibiotics are designed to kill bacteria, and while they target harmful species causing infections, they often kill beneficial bacteria as well. After a course of antibiotics, it can take weeks or months for your gut microbiome to fully recover, and sometimes the composition never quite returns to its previous state. During this recovery period, you may be more vulnerable to digestive problems and infections. As you rest quietly, trillions of bacteria throughout your body continue their work. In your intestines, they are breaking down the remnants of your last meal, fermenting fibers, producing vitamins, and communicating with your immune cells. On your skin, they form invisible barriers against invaders. In every inhabited corner of your body, they persist, these ancient companions, these microbial partners who share your journey through life, invisible but indispensable, working not for you but alongside you in a partnership neither party fully controls but both depend upon. Beyond the bacteria that inhabit animal bodies, there exists another remarkable partnership, one between certain bacteria and plants, a relationship so essential that much of Earth's agriculture depends upon it. This is the story of nitrogen fixation, a quiet process that occurs underground, invisible to our eyes yet vital to nearly every ecosystem on the planet. Nitrogen is abundant in our atmosphere, making up approximately 78% of the air you breathe. Yet despite this abundance, most organisms cannot use atmospheric nitrogen directly. The nitrogen gas in air consists of two nitrogen atoms bonded together so tightly, in a triple bond so strong that breaking it apart requires enormous amounts of energy. Plants need nitrogen to build proteins, DNA, and chlorophyll, but they can only absorb it in the form of ammonia or nitrate, not as nitrogen gas. This presents a fundamental challenge. Nitrogen is everywhere yet largely unavailable. Enter the nitrogen-fixing bacteria, microbes that possess a remarkable enzyme called nitrogenase, which can break the triple bond of atmospheric nitrogen and convert it into ammonia that plants can use. This process, nitrogen fixation, is one of the most important biochemical reactions on Earth, bridging the gap between the abundant but unusable nitrogen in the atmosphere and the usable nitrogen compounds that all living things require. Some nitrogen-fixing bacteria live freely in soil or water, working independently to convert atmospheric nitrogen. But the most efficient nitrogen fixers form intimate partnerships with certain plants, particularly legumes such as peas, beans, clover, and alfalfa. These symbiotic bacteria, called rhizobia, enter into an arrangement with their plant hosts that benefits both parties profoundly. The partnership begins when a legume seed germinates and sends its first root into the soil. The young plant releases chemical signals into the surrounding soil, compounds that rhizobia bacteria can detect. The bacteria respond by swimming through the water films between soil particles toward the root, drawn by the plant's signals like moths to a flame. When rhizobia reach the root hairs, the delicate single-cell extensions that increase the root's surface area. They attach to the surface and begin a carefully orchestrated process of infection. The word infection might sound harmful, but this is an infection by invitation. The root hair curls around the attached bacteria, and the plant's cells form an infection thread, a tunnel of plant material that allows the bacteria to enter the root's interior. The bacteria travel down this thread, moving from cell to cell, until they reach the root cortex, the inner tissue of the root. Here, the plant cells begin to divide rapidly, forming a special structure called a root nodule. Inside these nodules, conditions are carefully controlled. The plant provides the bacteria with carbohydrates produced through photosynthesis, a rich energy source the bacteria need to power the energy-intensive process of nitrogen fixation. The plant also produces a special protein called hemoglobin, which has a pink color and gives the interior of healthy nodules a distinctive appearance. Hemoglobin binds oxygen, keeping free oxygen levels in the nodule very low. This is crucial because nitrogenase, the enzyme that fixes nitrogen, is extremely sensitive to oxygen and ceases to function when exposed to it. Within this protected, oxygen-depleted environment, the rhizobia work continuously to convert atmospheric nitrogen into ammonia. The ammonia is immediately absorbed by the plant and incorporated into amino acids, the building blocks of proteins. In exchange for this nitrogen, the plant continues to supply the bacteria with carbohydrates, maintaining a steady flow of energy to fuel their work. This partnership allows legumes to thrive in nitrogen-poor soils where other plants would struggle. A single soybean plant might host hundreds of nodules on its roots, each one a tiny factory where bacteria and plant cells work together to capture nitrogen from the air. Over a growing season, a field of legumes can fix substantial amounts of nitrogen, enriching the soil not only for themselves but for subsequent crops planted in the same field. Farmers have known about this enriching effect for thousands of years, long before anyone understood the microbial mechanisms behind it. Ancient agricultural practices included crop rotation, planting legumes in fields one year and grains the next, allowing the soil to recover its fertility between grain crops. We now understand that this recovery was due to the nitrogen fixed by rhizobia in the legume roots, nitrogen that remained in the soil after the legume crop was harvested. The amount of nitrogen fixed by legumes and their bacterial partners is substantial. A field of alfalfa, one of the most efficient nitrogen-fixing crops, can fix 100 to 300 kilograms of nitrogen per hectare per year. This is nitrogen pulled directly from the atmosphere, converted into a form usable by plants, requiring no industrial fertilizer production. In an era when industrial nitrogen fertilizer production consumes vast amounts of energy and contributes to environmental problems, the natural nitrogen fixation performed by bacteria represents a sustainable alternative that agriculture increasingly seeks to enhance and utilize. The specificity of the rhizobia-legume partnership is remarkable. Different species of rhizobia associate with different legume species, and the chemical signals exchanged between them are precise, ensuring that compatible partners find each other. A bacterium that forms nodules on soybean roots may not be able to colonize alfalfa roots, and vice versa. This specificity has evolved over millions of years, refining the partnership into a highly efficient system. When you walk through a field of clover or peas, or when you see alfalfa growing along a roadside, you are witnessing this ancient partnership in action. Beneath the surface, in nodules you cannot see, bacteria are converting atmospheric nitrogen, performing a service that makes agriculture possible, that feeds billions of people, that sustains ecosystems around the world. It is a quiet process, requiring no intervention, no technology beyond what evolution has provided. Yet its importance to life on Earth can hardly be overstated. As your breathing slows and deepens, imagine these underground nodules scattered throughout the soil wherever legumes grow. Inside each one, bacteria work steadily, breaking the strongest bond in nature, converting the inert nitrogen of the atmosphere into the building blocks of life. It is work that requires no light, no awareness, no intention, only the persistent action of enzymes refined over countless generations, only the continuation of a partnership that predates human civilization, that will continue long after we are gone, as long as legumes grow and rhizobia persist in the soil beneath them. Among all the remarkable abilities bacteria possess, few capture the imagination quite like bioluminescence, the production of light by living organisms. While we often associate glowing creatures with fireflies or deep-sea fish, some of the most abundant light-producing organisms on Earth are bacteria, microscopic beings that generate their own cold light through chemical reactions occurring within their tiny cells. Bioluminescent bacteria are found primarily in marine environments, inhabiting the open ocean, coastal waters, and the bodies of certain marine animals. The light they produce is typically blue-green, with wavelengths around 490 nanometers, a color that travels well through seawater and appears as an ethereal glow in the darkness of the ocean. Unlike the light from a flame or an electric bulb, which produces heat as a byproduct, bacterial bioluminescence is almost entirely cold light, with nearly all the energy of the reaction converted directly into photons rather than being lost as heat. The chemistry behind this glow involves a protein called luciferase and a small molecule called luciferin. When luciferase catalyzes the oxidation of luciferin in the presence of oxygen, the reaction produces light, along with an oxidized form of luciferin that must be recycled back to its original state before it can produce light again. The bacterial cells maintain steady supplies of these molecules, and as long as oxygen is present, the reaction continues, creating a sustained glow rather than a brief flash. What makes bacterial bioluminescence particularly fascinating is that individual bacteria do not glow alone. Instead, they coordinate their light production through a process called quorum sensing, a form of chemical communication that allows bacteria to detect how many of their kind are nearby. Each bacterium continuously releases small signaling molecules into its environment. When bacterial population density is low, these molecules diffuse away and remain at low concentrations. But when many bacteria are present in a confined space, the signaling molecules accumulate to high concentrations. When the concentration of these signaling molecules reaches a threshold, indicating that a quorum has been achieved, the bacteria respond by activating the genes responsible for light production. Suddenly, all the bacteria in the population begin glowing together, creating a visible light where moments before there was only darkness. This coordinated response makes evolutionary sense. The light produced by a single bacterium would be too faint to serve any purpose, but the combined glow of millions of bacteria can be bright enough to serve various functions. Some bioluminescent bacteria live freely in seawater, particularly in nutrient-rich areas where dead organic matter accumulates. These free-living bacteria can create spectacular displays when disturbed, a phenomenon known as milky seas or mareal. Sailors have reported sailing through vast stretches of ocean that glowed with an eerie blue-green light, caused by countless trillions of bioluminescent bacteria responding to the disturbance of the ship's passage. These glowing seas can extend for thousands of square kilometers and persist for days, creating one of nature's most surreal spectacles. Other bioluminescent bacteria form symbiotic relationships with marine animals, particularly certain species of fish and squid. These animals provide specialized light organs where bacteria can live and grow, supplying them with nutrients and oxygen. In return, the bacteria produce light that the host animal can use for various purposes: attracting prey, finding mates, confusing predators, or camouflaging themselves against downwelling light from the surface when viewed from below. The Hawaiian bobtail squid hosts one of the most studied bacterial bioluminescent symbioses. Each evening, as the squid emerges from the sand where it spent the day buried, it relies on light produced by Vibrio fischeri bacteria living in a specialized organ in its body cavity. The squid adjusts the intensity of this light to match the moonlight and starlight filtering down through the water, effectively erasing its shadow and making it nearly invisible to predators looking up from below. Each morning, the squid expels most of its bacterial population and retreats to the sand, and the bacteria that remain multiply throughout the day, preparing for the next night's light show. The anglerfish, those bizarre deep-sea predators with bioluminescent lures dangling in front of their mouths, also rely on bacterial light. The glowing lure, which attracts smaller fish close enough to be captured, contains colonies of bioluminescent bacteria that the anglerfish maintains and cultivates. In the perpetual darkness of the deep ocean where sunlight never penetrates, this bacterial light may be the only illumination for kilometers in any direction. Not all bioluminescent bacteria live in the ocean. Some species inhabit terrestrial environments, though they are far less common than their marine cousins. Certain bioluminescent bacteria have been found on decaying wood in forests, creating ghostly glowing patches that have inspired folklore about fairy lights and will-o'-the-wisps. These glowing logs and stumps occur when bacteria colonize the moist, nutrient-rich wood and reach population densities high enough to trigger coordinated light production through quorum sensing. The evolutionary advantage of bioluminescence varies depending on the bacterial species and its lifestyle. For symbiotic bacteria, the benefit is clear. By providing light to their host, they secure a protected environment and steady food supply. For free-living bacteria, the advantages are less obvious and remain a subject of scientific investigation. Some researchers suggest that the light might attract larger animals to areas where bacteria are concentrated, and these animals might then disturb the area and release nutrients that the bacteria can use. Others propose that bioluminescence might be a byproduct of other metabolic processes rather than an adaptation specifically for producing light. Whatever the evolutionary explanation, the result is undeniably beautiful. Living light produced by organisms too small to see individually, glowing in the darkness of the ocean or the forest floor. It is light without fire, cold photons emerging from chemical reactions refined over millions of years of evolution, a reminder that even the smallest life forms can illuminate their surroundings. As you lie in the quiet darkness, imagine the ocean depths where bioluminescent bacteria glow within the bodies of squid and fish, or the coastal waters where billions of bacteria create patches of light in the waves. Imagine them coordinating their glow through chemical signals, waiting until enough of them are present before switching on their light-producing genes, creating together what none of them could create alone. It is cooperation at the microscopic scale, communication through molecules and the production of light from life itself, all occurring constantly in the hidden corners of our planet, unseen by most but perpetually present, a gentle luminescence in the dark. When we speak of microbes, we often focus on bacteria, but the microscopic world contains far more diversity than any single group can represent. Microorganisms span multiple domains and kingdoms of life, each with unique characteristics, evolutionary histories, and roles in the ecosystems they inhabit. Let us take a quiet journey through this diversity, exploring the major types of microbes that share our world. We have spent much time discussing bacteria, those prokaryotic cells without nuclei, whose simplicity and adaptability have allowed them to colonize virtually every environment on Earth. Bacteria are perhaps the most abundant and diverse group of microbes, with millions of species estimated to exist, though only a tiny fraction have been formally described and studied. They fill countless ecological roles: decomposers breaking down dead organic matter, photosynthesizers producing oxygen, pathogens causing disease, and mutualists forming partnerships with plants and animals. But bacteria are not alone in the prokaryotic world. They share it with the archaea, microorganisms that superficially resemble bacteria in size and structure but differ profoundly in their biochemistry and evolutionary origin. Archaea were once thought to be simply unusual bacteria, but molecular studies revealed them to be as different from bacteria as they are from the cells that make up your body. They constitute an entire domain of life, one of the three major branches on the tree of life, alongside bacteria and eukaryotes. Many archaea are extremophiles, thriving in environments that even hardy bacteria find challenging. The methanogens, archaea that produce methane as a metabolic waste product, live in oxygen-free environments like swamps, the guts of cattle and termites, and deep-sea sediments. The halophiles we mentioned earlier, loving salt concentrations that would desiccate most life, are primarily archaea. The thermophiles and hyperthermophiles living in the hottest environments on Earth are often archaea as well. The cell membranes of archaea differ chemically from those of bacteria, with unique lipids that help them survive in extreme conditions. Their cell walls, when present, lack peptidoglycan, the tough polymer that gives bacterial cell walls their strength. Many of the enzymes and molecular machines inside archaeal cells more closely resemble those of eukaryotes than those of bacteria, suggesting a closer evolutionary relationship between archaea and the complex cells that make up plants, animals, and fungi. Speaking of fungi, these organisms represent the first group of microbes that belong to the eukaryotes, organisms whose cells contain nuclei and other membrane-bound compartments. While many fungi are large enough to see with the naked eye, such as mushrooms and molds, the fungal kingdom also includes numerous microscopic species, particularly the single-celled yeasts. Yeasts are perhaps the most familiar microscopic fungi, known for their roles in baking bread and fermenting beer and wine. A yeast cell is substantially larger than a bacterial cell, typically 5 to 10 micrometers in diameter, and its internal structure is far more complex. Inside the cell membrane lies a true nucleus containing the yeast's genetic material, surrounded by its own double membrane. The cytoplasm contains mitochondria, the organelles that produce energy through aerobic respiration, and various other structures that bacteria and archaea lack entirely. Yeasts reproduce in several ways, including budding, where a small outgrowth forms on the parent cell and gradually enlarges until it separates as a complete new cell. Some yeasts can also reproduce sexually, fusing with other yeast cells to exchange genetic material before dividing again. This sexual reproduction allows for genetic recombination and adaptation in ways that are more complex than the simple binary fission of bacteria. Fungi, including microscopic species, play crucial roles as decomposers in ecosystems. They secrete enzymes that break down complex organic materials like cellulose and lignin, the tough structural polymers and plant cell walls that few other organisms can digest. In forests, fungal decomposers are essential for recycling nutrients from dead leaves and wood back into forms that plants can absorb and use. The fourth major group of microbes consists of protozoa, single-celled eukaryotes that are generally more complex and larger than bacteria, archaea, or even most yeasts. Protozoa are remarkably diverse, including species that swim using hair-like cilia that beat in coordinated waves, species that move with whip-like flagella, and species that crawl and engulf food particles using temporary extensions of their cell membrane called pseudopodia. Many protozoa are predators, feeding on bacteria and smaller microbes in aquatic environments and soil. A single drop of pond water might contain dozens of protozoan species, each pursuing its own strategy for finding food and avoiding becoming food for larger predators. These microscopic hunters play important roles in controlling bacterial populations and cycling nutrients through aquatic food webs. Some protozoa form symbiotic relationships with larger organisms. The protozoa living in the guts of termites, for example, produce enzymes that break down the cellulose in wood, allowing termites to extract nutrition from their diet of timber. Without these protozoan partners, termites could not survive on wood alone. What unites all these diverse microbes, despite their profound differences in structure and biochemistry, is their small size and their fundamental importance to the functioning of Earth's ecosystems. They are the foundation of nearly every food web, the recyclers of nearly every ecosystem, and the invisible partners without which most larger organisms, including humans, could not survive. The diversity we see among microbes today represents billions of years of evolution, countless experiments in how to survive and reproduce in a competitive world. Each group has found its own solutions, its own biochemical strategies, its own ecological niches. Bacteria achieved success through simplicity and rapid reproduction. Archaea carved out the most extreme environments, places where competition from other organisms is minimal. Fungi developed the enzymatic tools to break down the toughest organic materials. Protozoa grew larger and more complex, becoming the predators and grazers of the microbial world. Together, these microbes create an invisible world of staggering complexity and beauty, a realm where the fundamental processes of life play out on scales too small for us to perceive directly, but too important for us to ignore. As you rest in the stillness, this microbial world continues its ancient patterns, each organism pursuing its own survival, each group contributing to the greater whole, all of them together forming the living foundation upon which the visible world ultimately rests. We have traveled together through the invisible realm of microbes, from the ancient stromatolites that record the earliest chapters of life on Earth, to the bioluminescent bacteria glowing in ocean depths, from the extremophiles thriving in boiling water and frozen ice to the trillions of bacteria quietly working within your own body. It is time now to rest in the quiet understanding of what these tiny organisms mean for our world and for our lives. Microbes are everywhere. This is not metaphor or exaggeration, but literal truth. They exist in the air you breathe, in the water you drink, in the soil beneath your feet, on every surface you touch, and throughout your body from your skin to the deepest reaches of your digestive tract. They float in clouds, helping water droplets form around their tiny bodies. They live kilometers beneath the ocean's surface and kilometers below the ground, in rock pores and underground aquifers. They persist in the driest deserts and the coldest glaciers, in acidic hot springs and alkaline soda lakes. This omnipresence is not something to fear, but rather something to marvel at quietly. Most microbes are harmless to humans, and many are actively beneficial, contributing to our health, helping us digest food, producing vitamins, training our immune systems, and protecting us from the few species that might cause harm. The relationship between humans and microbes is not one of opposition, but of coexistence, a partnership that has evolved over millions of years as our ancestors and their microbial companions adapted to one another's presence. Consider the remarkable fact that microbes have been present on Earth for roughly 3.5 billion years, while humans in our modern form have existed for only a few 100,000 years. Microbes witnessed the formation of our atmosphere, the oxygenation of our planet, the rise and fall of countless species, the movement of continents, and the cycling of elements through rock and water and air across unimaginable spans of time. They are the elders of our planet, the organisms with the longest continuous presence, the witnesses to nearly every chapter of Earth's biological history. Their contributions to life on Earth extend far beyond their own existence. Every breath of oxygen you take was produced by photosynthetic organisms, many of them cyanobacteria, continuing the work their ancestors began billions of years ago. The nitrogen in the proteins that make up your muscles and organs was likely fixed from the atmosphere by bacteria working in partnership with plants. The nutrients cycling through ecosystems, moving from soil to plants to animals and back to soil, are processed and transformed by countless microbial decomposers, breaking down complex molecules into simpler forms that other organisms can use. The speed at which bacteria can reproduce, doubling their numbers every 20 minutes under optimal conditions, gives them an extraordinary capacity to adapt to changing environments. This rapid reproduction means that beneficial mutations can spread through bacterial populations in days or weeks, allowing bacteria to evolve solutions to new challenges with a speed that larger organisms cannot match. It is both a reminder of their resilience and an explanation for their success in colonizing virtually every habitat on Earth. Even in environments that seem utterly hostile to life, extremophiles persist, redefining our understanding of where life can exist and what conditions it can tolerate. These bacteria, living in boiling water and frozen ice, in acid and radiation, demonstrate that life is not fragile but remarkably tenacious, not rare but almost impossibly persistent. They expand the boundaries of the possible and make us reconsider assumptions about the limits of life, not only on Earth but potentially on other worlds as well. The partnerships microbes form, whether with plants exchanging nitrogen for carbohydrates, with squid providing light for camouflage, or with humans helping to digest food and maintain health, reveal that cooperation is as fundamental to life as competition. These symbioses, refined over evolutionary time, show that survival often depends not on domination but on mutual benefit. Not on isolation, but on connection. As you settle deeper into rest, let your awareness of this microbial world remain gentle and calm. There is no need to do anything with this knowledge, no action required, only perhaps a quiet appreciation for the invisible processes that surround and sustain all visible life. The bacteria in your gut are working steadily, breaking down the food you consumed hours ago, producing vitamins and protective compounds. The bacteria on your skin form invisible barriers against potential invaders. In soil and water around the world, microbes continue their ancient work of recycling, fixing, decomposing, and transforming. The microbial world asks nothing of you and continues regardless of your awareness of it. It operated for billions of years before humans evolved and will persist long after we are gone. Yet understanding it, even in this gentle and incomplete way, offers a different perspective on your place in the living world. You are not separate from nature, but deeply embedded within it. Not a solitary organism, but a walking ecosystem. Not independent, but connected through countless invisible threads to the microbial foundation that underlies all life on Earth. Let this understanding be a source of quiet comfort rather than concern. In the darkness and stillness, as your breathing slows and your thoughts drift towards sleep, the microscopic world continues its patient work. These ancient organisms pursuing their simple imperatives of survival and reproduction, together creating the complex web of interactions that we call life. If you found peace in this exploration of the invisible world, if these sleepy science facts about microbes brought you some measure of calm or wonder, consider subscribing to Nighttime Science for more quiet journeys through the realms of biology, physics, and the natural phenomena that surround us. A gentle like helps others discover these soothing explorations of science designed for rest and sleep. For now, let your mind rest with the microbes, those smallest citizens of Earth. Those invisible companions who have been here since the beginning and will persist far into the future, quietly sustaining the world we know. Sleep well.